皆様、本日、バイオセトジのウェビナーご参加いただき、誠にありがとうございます。私、バイオセトジン前臨床 CRO サービス事業部、長塩と申します。本日はどうぞよろしくお願いいたします。本日のウェビナーでは、近年非常に注目を集めている創薬モデラティであり、T 細胞エンゲージャーをテーマに、その開発を支えるためにどのように適切な動物モデルを選択すべきかについて、ユージェリオ博士よりご講演いただきます。レオ博士は、甲冑夏季大学薬学部にて卒業後、北京大学薬学部にて博士号を取得されておりました。その後、イリノイ工科大学及びノースウェスタン大学にてシニアリサーチャーとして勤務され、主要ナノメディシンにおける血液の関門、デリバリや肺障害発症メカニズムなどの研究に従事されておりました。2020年にバイオセトジンへ入社後、薬理リソース及び製品部門においてシニアディレクターやマネージャーを歴任され、現在は薬工評価部門のディレクターを務めていらっしゃいます。この役割において、劉博士には、BioMice事業システムを統括し、300種類以上の PDX モデルを含む標準化されているの薬効評価リソースデータベース及びインテリジェントデータサイエンスシステムの構築を主導されておりました。また、これまでに国内外の多くの製薬企業プロジェクトを手掛け、腫瘍抗体薬、免疫再構築腫瘍モデルに用いて薬効評価、血液の関門透過、腫瘍及び自己免疫疾患領域、さらにヒト化マウスを用いた PDX 応用研究など幅広い分野において豊富な経験をお持ちです。本日のウェビナーでは T 細胞エンゲージャーに関する最新の業界動向を腫瘍及び自己免疫疾患の領域からご紹介するとともに、ヒト化マウス、免疫再構築マウス、各種疾患モデルなどの動物モデルが TCE 開発のどの段階でどのように活用するべきか、さらに適切なモデル選択のための戦略及び重要な評価ポイントについて詳しくご説明いただきます。なお、Q&A ボックスにすでにオープンしておりますので、セッション中いつでもご質問をお送りください。いただいたのご質問には、後ほど弊社担当よりメールご回答いたします。それでは早速でございますが、劉博士にハンドをお渡しいたします。どうぞよろしくお願いいたします。
All right, it looks like we are ready to begin. So hello again and welcome to BioSaturn's webinar series. It's nice to meet you all online. So in our session today, we will be talking about a very popular drug modality, which is the T-cell engagers. And we will be discussing how to choose the right models to support their development. So before we dive in, let me introduce our team here. So just a little bit about myself. My name is Yuan Tian, and I am the content manager at BioSaturn Boston Corporation. I will be your moderator for today's webinar. And I'm also joined by two amazing colleagues from the BioSaturn team who will be serving as our panelists. So with us today are Dr. Hannah Horton, Regional Director of Business Development, and Dr. Lucy Peterson, Business Development Manager at BioSaturn. So thank you both for being here. We are really excited to hear your perspectives. And next, it's my pleasure to introduce Dr. Yujie Liu will be our speaker for today's webinar. So Dr. Liu received her bachelor's degree from the School of Pharmacy at Huazhou University of Science and Technology and her PhD from the School of Pharmacy at Peking University. She then served as a senior researcher at the Illinois Institute of Technology and at Northwestern University, where she focused on areas such as blood-brain barrier delivery for tumor nanomedicine and the mechanisms of lung injury. And in 2020, Dr. Liu joined BioCytogen, where she successively held positions as Senior Director and Manager of the Pharmacological Resources and Product Department. She is currently the Director of the Pharmacology and Pharmacodynamics Department, and in this role, she leads the BioMice business system, and she's played a key part in building a standardized pharmacodynamic resource database that now includes more than 300 PDX models, along with an intelligent data service system. And over the years in BioCytogen, Dr. Liu has managed many projects for pharmaceutical companies, both domestic and international, and her expertise spans tumor antibody drugs, immune reconstituted tumor efficacy, blood-brain barrier penetration, oncology, autoimmune diseases, and PDX applications using humanized models. So for today's webinar, Dr. Liu will share recent industry insights, including both oncology and autoimmune applications of TCE, and she will also discuss when and how animal models, including the humanized mice, the immune reconstituted mice, and disease models can be used in this field, as well as strategies and case studies for selecting the right models to support T-cell engager development. Okay, we're almost there. Just a quick reminder, the Q&A box is already open, so feel free to send in your questions anytime during the session, during the presentation. We will save some time at the end to answer as many questions as we can. All right, without further delay, I will hand things over to Dr. Liu.
Yeah, hello everyone. I'm very glad to be here. And today my topic is model selection in preclinical studies of bispecific antibodies, which is T-cell engager from tumors to autoimmune hotspots. So today I will more focus on the in vivo models for the efficacy evaluation in both tumor and autoimmune disease models, as well as PK studies. And meanwhile, we want to discuss about toxicity of T-cell engager and how to evaluate using different kinds of mouse models. So up to now, there are 19 globally approved bispecific antibodies, and among them, 10 of them are T-cell engagers. And we set up a list here. We can see that they are targeting different kinds of TAA target, and the indications of these kinds of drugs are solid tumor and blood tumor. And nowadays, there are also some T-cell engagers are in clinical stage for the autoimmune diseases. And we can also take a look at the bispecific antibodies, the CD3 antibody part. And we can see that OKT3 and SP34 is very hot in this T-cell engager format. SP34 is the most popular one because it can also react with the monkey protein. So that's why it is really popular. But there is also a problem of SP34 is that it has the CRS problem. So talking about the characteristic of T-cell engager, they have a lot of advantages as well as they have some disadvantages. So as to the advantages, they have potent anti-tumor activity and high efficacy at a very low dosage, and it has broad applicability. For example, for blood tumor, solid tumor, and autoimmune diseases treatment. And it has a very good engineering flexibility, and is kinds of off-the-shelf drugs for the evaluation, not like the CAR T cells. And there is also a lot of T-cell engagers that are trying to target novel targets like peptides presented by MHC. So as to the disadvantage of T-cell engagers, the most common risk is about CRS. And another thing is about on-target of tumor toxicity that is caused by the expression of the target in the normal tissue. And several of the T-cell engager that has been approved also have the half-life problem. It has very short half-life, so it is not very convenient for the patient's treatment. And another thing about T-cell engager is about the tumor microenvironment suppression and the resistance or escape of the tumor treatment. And meanwhile, there is also narrow therapeutic windows. So based on the advantages and disadvantages of T-cell engagers, there are coming up a lot of kinds of formats of T-cell engagers. So we are also trying to summarize what kinds of models are best for the T-cell engager evaluation. So we first make a summary of the T-cell model selection, basically based on the in vivo efficacy, PK evaluation, and the safety evaluation. For the in vivo efficacy, the two main indication is tumor and autoimmune disease. And for the tumor part, we can use immune reconstitution model, including PBMC and HSC reconstitution model, combined with CDX or PDX model. And we can also use humanized mice in the and plus TA humanized cell line for the tumor efficacy evaluation. For the autoimmune disease models, we can first use the humanized mice for the PD evaluation, and we can also combine different mice with disease models. For example, rheumatitis, arthritis, and SLE and EAE model for the evaluation. For the PK part, usually we can use the one we use for the in vivo evaluation, and we can also try to use FcRn humanized mice, which is better for the antibody that has been modified in their Fc part. And for antibody, we can also use ALB FcRn double humanized mice. And for the safety evaluation, the most two important part is CRS and off-target toxicity. In this case, humanized mice are the best choice for the evaluation. So we can see that humanized mice appears in every part of the study. So today we will more focus on the humanized mice. And meanwhile, we also discuss about other models, and we can also compare which one is the best. As to the tumor models, we can use genic model, which is human target and checkpoint humanized mice and humanized cell lines for the evaluation. And we can also use xenograft model plus immune reconstitution models for the evaluation. And if you want to take a challenge, we can also try to use orthotopic and metastatic tumor models for more indications development. So talking about CD3-EDG humanized mice. We want to let you know the targeting strategy and the target expression first. What we do is to modify three genes. The three of them are CD3E, CD3D, and CD3G. So it is like a triple humanized mice. This allows us to makes us to be able to evaluate different kinds of CD3 binders. They can target in each other part of the CD3 and protein on the T cells. For the validation, we validate the humanized mice both in the mRNA level and the protein level. And we also did some validation of the mice in vitro. So first, we give the mice most of human CD3 and the CD20 antibodies. And we can see that it has a comparable humanization levels compared to wild-type mice. And we detected L2 and F and gamma at different time points, so which we got very good result using the humanized mice. And meanwhile, we also compare the wild-type mice with the humanized mice after the OVA immunization. And we can see that the secretion of the different subtype of antibodies are comparable in both wild-type mice and the humanized mice. And we also did some in vivo efficacy evaluation to validate if the mice works well. So for this study, we inoculate MC38 into the CD3-EDG humanized mice. We can see that the right curve shows the result of mouse PD-1 treatment, and which we can see a very good anti-tumor efficacy, which means this model works good for the immune checkpoint antibody. And meanwhile, we give them human CD3E antibody, and the tumor grows even faster than the control group, which means which is caused by the AICD, which is activation induced cell death. And we can also detect the T cells in the peripheral blood and the tumor the T cells infiltrated in the tumor, and we can see the decrease of the T cells both in blood and tumor. So this mice works good both in vitro and in vivo. So based on the good full validation of the mice, we have done quite a lot of studies using these kinds of humanized mice in solid tumor, blood tumor, and autoimmune disease models, and based on different kinds of targets. And we have done quite a lot of studies for different purposes. So today I would like to show you several cases in different disease areas. So first one is for the tumor treatment. In this study, we give the CD3 EDT humanized mice inoculated with BCMA humanized MC38. We give the GR1803, which targets BCMA and CD3E for the treatment. And we can see a dose-dependent manner, and which means this mice is very good for the BCMA T cell engager. And another very hot target in these 2 years is DLL3 bispecific antibody. FDA approved the first DLL3 T cell engager last year, and that's why nowadays there is a lot of consults and a lot of projects going on for these kinds of models. So in these studies, we use DLL3 humanized B16F10 cell lines inoculated into the CD3 EDT humanized mice. And we give the AMG 757, and we can see a very good anti-tumor efficacy after the treatment. And another very hot target is CD20, and glofitamab is also approved drug for the tumor treatment. So in these studies, we inoculate CD20 humanized MC38 into CD20 CD3 EDG double humanized mice, and we give 2 different dosage of glofitamab, and we can see a dose-dependent manner. And meanwhile, the B cells didn't decrease a lot, and there is no significant difference between the control group and the treatment group, which means that the test articles are mainly distributed in the tumor area, and the T cells decreased after the treatment, which is mainly caused by the AICD of the test articles. And so based on different test articles and based on their different targets, we have generated a series of different TAA and CD3 EDT humanized mice for the efficacy evaluation. And meanwhile, we have also generated a series of TAA humanized cell lines to inoculate in different kinds of humanized mice for the anti-tumor efficacy evaluation. So we have done a lot of studies, and we also encountered some problems for the evaluation. In this study, first we generated HER2 humanized MC38, and we inoculate this cell line into CD3E HER2 double humanized mice. And unfortunately, we didn't get a very good anti-tumor efficacy at different dosage. So we had some internal discussion why it didn't work. And there are several possibilities. One is the specificity and affinity of the binding sites of CD3 antibody. And second one may be the loss of target antigen expression after the inoculation of the tumor or after the treatment. And the third one may be the formats of the bispecific antibody. So we did what we can do to modify these models. So we generated another cell line, which is HER2 high expression MC38. This cell line has a comparable expression level of human cell line, which is NCIN87. And the orange curve is the original version, which is HER2 low expression MC38. Using the high expression version, we give the treatment of HER2 CD3 T cell engager, and we found a very good anti-tumor efficacy, and which is also shows a very good dose-dependent manner. So this have give us some suggestions that for the evaluation of your test articles, we may consider the affinity of the test articles to the TAA, and meanwhile we have to consider which level of TAA we need for the evaluation and what kinds of patients we are going to treat for your future studies. Another very hot models used is immune reconstitution model, and this is most traditional ones that's used in the pharmaceutical areas. The one is PBMC reconstitution, and the other is HSC reconstitution. For PBMC reconstitution, we can mostly we can get T cells after the reconstitution, so which makes them a very good model for the evaluation of T cell engager. And another one is HSC reconstitution model. For this kind of model, it has B cells, T cells, NK cells, and memory cells, depending on what kind of mice you are going to use. And as long as it has B cells, it is also a very good model for the PD evaluation of targets that are targeting autoimmune disease models. So we give one example using PBMC reconstitution. We give the PBMC reconstitution model the HER2 T cell engager in both CDX model and PDX model. We can see that it shows a very good anti-tumor efficacy after the treatment. Talking about PBMC reconstitution, the most severe problem is GVHD symptoms after the inoculation of PBMC. GVHD generally happens 4 weeks after the reconstitution. So to get avoid of the influence of GVHD, we generate MHC1 and MHC2 double knockout mice. This allows us to extend the lifetime of the mice and meanwhile prolong the time, prolong the treatment window for the study. We can see that the mice can survive more than 100 days post engraftment of PBMC. And for the GVHD score, it comes up 42 days. And the mice can, meanwhile, the mice can also get a very good reconstitution levels after the PBMC reconstitution. And we also use this mice for the HER2 T cell engager evaluation, and we can see it has a very good anti-tumor efficacy. And even we give them some test articles, the body weight of the mice still increased during our study. So it won't for some other kinds of PBMC reconstitution models, when we give them treatments, the body weight of the mice generally will decrease. So at this time point, someone will ask, there are humanized mice and there are immune reconstitution mice, which are the best for the T cell engager experiment. So we also did some internal work in our company. So we used DLL3 T cell engager for our internal study. We generated 4 formats of the T cell engager, which target DLL3 and CD3. We use syngentic model and the xenograft model for the evaluation. For the syngentic model 1, we use DLL3 humanized B16F chain inoculated into the EDG humanized mice. And for the xenograft model, we use PBMC reconstitution and inoculated with SHP77. Which has a high expression level of DLL3. In these 2 models, we found out that format 3 has the best anti-tumor efficacy, and for the positive control, it also have a very good anti-tumor efficacy. At the end of the study, we made some summarization of the two and comparison of the two apparent kinds of models. We also use different donors for the evaluation of SHP77. And we found that for FORMA2 and FORMA3, the syngentic model and xenograft model both shows very good anti-tumor efficacy. And for FORMA1, they showed good anti-tumor efficacy in syngentic model, and for xenograft model, we didn't find very good anti-tumor effect. So these two models looks like they are kinds of translationable, but we are also collecting more data for the further evaluation and to make a more solid summary. And meanwhile, apart from the TAA T-cell engager, there are also TCRM T-cell engager. So based on that, we also generated a series of HLA humanized cell lines in C50 background. And meanwhile, we also generated HLA humanized mice in B6 and BNDG background. And meanwhile, we also generated different humanized HLA and peptide humanized CDX models. That's all for the tumor part. Next, I would like to talk about autoimmune disease models. Up to now, there are a lot of companies that has been used their T-cell engager for the evaluation of autoimmune diseases. And among them, CD19, CD20, BCMA are the most popular one for the evaluation. It is because the abnormal B cells are involved in the pathogenesis of various autoimmune diseases, and the depleting B cells can potentially elevate the autoimmune disorders. For the evaluation of these kinds of T-cell engager, we can first do some PD evaluation, and this is to use different kinds of mice and just without any modeling, we just give them test articles, and we can try to detect the B cells in the mice if they can be depleted after the treatment. And next, we can do the efficacy evaluation. We can use the humanized mice and autoimmune disease model induction method to induce the autoimmune symptoms, and then give the treatment. So first, I would like to show you a PD evaluation for the in vivo B cell depletion of T-cell engager. In these studies, we give the mice glofitamab, and we can see that there is a significant decrease of the B cells after the treatment. And the T cells first increase and then decrease, which is AICD symptoms. Biocytogen also have a very good autoimmune disease platform. So we have also generated models that related to B cell function, like CIA-induced RE model, MOG-induced EAE model, and Pristin-induced SIE model. Today, I would like to show you one model that has been validated using our humanized mice. In these studies, we use MOG 1 to 125 to induce EAE. And we can see the blue curve shows a very good induction of EAE after the immunization. And we also give glofitamab to the mice for this kind of treatment. And the score of the treatment group is kind of like 0 after the treatment, which means that this glofitamab can be used for treat EAE model in preclinical studies. And we also detect the B cells and T cells in the blood. And we can see after the treatment, the B cells has almost been depleted after the treatment. And for the T cells, it will first increase and then decrease after the treatment. And 28 days, the T cells comes back and it continues to grow, to increase after we draw the treatment. And we can also see the B cells in the spleen was depleted, and the T cells is kind of comparable to the modeling group. Next is about the PK studies. So today I will more focus on the FcRn humanized mice, because for a lot of T cell engagers, they do not have a very good half-life. So there is a lot of work to be done to prolong the half-life of the T cell engager. So for this kinds of evaluation, we will more suggest the FcRn humanized mice, because it better access the pharmacokinetics of human therapeutics, especially for the antibody that has been modified by the in the Fc region. And for other kinds of antibodies, that's like the nanobodies, they have to be they usually bind to the albumin in the blood to prolong the half-life of their test articles. So we in this kinds of antibody studies, we generally suggest ALB FcRn double humanized mice for the PK evaluation. Next, I would like to discuss about the toxicity of T cell engager. So it is reported that there are mainly 4 types of toxicity induced by T cell engager. One is CRS, that is cytokine release syndrome, which is caused by the reaction of the T cells. And meanwhile, there will be neurotoxicity symptoms, which is also caused by the cytokine-driven BBB disruption or CNS infiltration. Meanwhile, there is on-target off-tumor toxicity. It is because antigen can also sometimes also expressed on the normal cells, which leading to the killing of healthy tissues. And some other immune-related toxicity happens in the after the treatment. So in this case, if we want to evaluate both CRS and on target of tumor efficacies, humanized mice is the best one for the evaluation, because it also have the expression of the antigen in the mice itself. So today I would like to give you a study that we have done using CD20, CD3E double bispecific antibody. We know that there are several CD20 T cell engager, and today we want to compare mosunetuzumab and glofitamab. We know that the binding mode of the 2 antibodies is different, and they also have different Fc part and binding affinity. And it is reported that glofitamab has a higher accuracy of DRS after the treatment compared to mosunetuzumab. So we give the test articles to the double humanized mice. And we can also see that the blue column, which shows the data of glofitamab, has comparable, is a little higher than the mosunetuzumab. Which made them also a very good model, makes the humanized mice also a very good model for the evaluation of CRS. And we also detect the liver function and kidney function after treatment. We can also see that creatinine was increased after the 2 test articles treatment. I would like to let you know that there is a trend to use humanized mice for the toxicity study, and it is widely used in pharmaceutical companies, especially for FDA and NMPA and applications. And up to now, 15 drugs candidates has received an IND approval from NMPA, including CD3E. T cell engager, and also 5 drugs candidates received IND approval from FDA, also including CD3E. So at the last, I would like to discuss about the next generation of T cell engager and what kinds of model we can use. Here is a whole strings of T cell engager. So they are more focusing on solid tumor, and they are they are trying to improve the safety as the CRS is quite common after the treatment. And nowadays, there are more multi-specific, logic-gated, tri-specific, or higher order T-cell engager coming up. And a lot of clients are also trying to discover novel antigens, and they also want to overcome the immunosuppression, and they also want to do a lot of work for the PK improvement. And meanwhile, they also want to use combination therapies to improve the anti-tumor efficacy. Talking about the tri-specific or multi-specific antibodies, generally there are two strategies. One is targeting two tumor-associated antigens to prevent tumor escape by antigen loss. So, for example, they can target BCMA, GPCR5D for the CD3 T-cell engager for the tumor treatment. Another strategy is targeting the two T-cell co-stimulatory receptors for optimal activation of the T-cells. For example, we can also use CD3, CD28, and HER2 for the tumor evaluation. So based on the need of our clients, we also generated a series of triple or even 4 target humanized mice for the evaluation. So another question will come up, how to choose the best combination of the TAA for the T-cell engager. So what we did is to do the single-cell RNA-seq of the PDX model. And for example, when we tap in EGFR and HER2 in our database, we found out that not all the PDX cells express both EGFR and HER2. So some of them only express EGFR and some of them only express HER2. We can also do the correlation between HER2 and EGFR, and they have a very good correlation in the tumor. And we can also choose the highest, the model that has the highest co-expression of the two targets. And we can also try to select which model best for the test article evaluation. And another method we use is to use immunofluorescence for the evaluation. In this study, we get a series of PDX model, and we stand 6 markers for each PDX model. So we can see like 15 combination of different targets. For example, we can see EGFR expression level and HER2 expression level, and we also have the merge of the two targets to find out the percentage of the co-localization of these two targets. Yeah, so that's the summary of our resources. We have BioMice resources, like we have a lot of humanized mice, different kinds of immunodeficient mice. We have rat and dogs. We have different kinds of disease models, and including tumor models and autoimmune disease models. And we have a very big detection technology platform for all of them to help our clients for their preclinical studies like PK, PD, toxicity, or mechanism studies. In the end, help them with IND application. So that's all for my topic. So I'm very glad to see if there is any questions.
本日、バイオセトジのウェビナーにご参加いただき、誠にありがとうございました。以上で、両博士により講演を終了となります。本日の内容につきましては、ご質問などがございましたら、info@biocetogen.com.cn までメールにてご連絡ください。弊社の担当者より、後ほど連絡させていただきます。また、こちらの QR コードをスキャンしていただけますと、バイオセトジの公式ウェブサイトにご覧いただけますので、ぜひご活用ください。改めて本日どうもありがとうございました。